Welcome to the new topic on topical formulations, and this topic is related to the bioequivalence requirements for the topical formulations. If you have worked or if you are working onto the formulations like gels, creams, ointments and many others which are applied onto the skin or which are inserted into the body cavities for local or systemic application, that time for some of the formulations the bioequivalence demonstration is required. If the formulation is containing the soluble API or dispersed API, that time you need to perform some studies to show equivalence with the reference formulation. The bioequivalence is the requirement, or you can say the regulatory requirement, the quality requirement for the generic formulations to demonstrate the therapeutic equivalency. If you have worked or if you are working onto the topical formulations, then you might have heard the terms like Q1, Q2, Q3 equivalence. You might have heard regarding the similarity and sameness of the qualitative and quantitative composition. You might have heard regarding the microstructural sameness. You might have heard regarding the IVRT and IVPT, clinical endpoint study, vasoconstriction studies and many more. So this video is an introduction to all these terminologies which are related to the demonstration of bioequivalence for topical formulations. Let's start with introduction. Topical formulations are the formulations like creams, gels, lotions, transdermal patches, ointments and many more. These are used in the dermal application, and these are the topical formulations in the dermatology. These are applied onto the skin and sometimes in the body cavities, mucous membranes. Considering the challenges of bioequivalence, the topical formulations are complex, and establishing the bioequivalence is more challenging for the topical formulations compared to the oral formulations, because there is a difference and complexity in the topical formulations which affects the drug release and the drug absorption, drug penetration or permeation. The application site is skin. There are variations in the skin, variations as per the body parts, and many more things are there which are showing the variability. Regulatory science is evolving to ensure the safe and effective generic topical formulations. From long time, the in vitro testing is getting evolved to show the similarity and to demonstrate the safety and efficacy of the dermal formulations. Now coming to the Q1, Q2 and Q3 equivalence for the topical formulations. Q1 is qualitative sameness of the excipients and components. Q1 sameness provides you the understanding about the qualitative composition, that is generic must contain the same components as per the reference formulation, and these components are API and the excipients. Then second is the quantitative sameness, that means the components excipients must be present in the same concentration as the reference formulation. And Q3 is the microstructural sameness. It refers to the physical and structural properties of the semi-solids. The microstructural sameness studies parameters are viscosity, globule size, phase distribution, rheology, and some others which affects the drug release, skin permeation, and the patient acceptability. regulatory importance of Q1, Q2, Q3. Q1, Q2 plus Q3 equivalence makes the generic formulation case stronger for granting or application of the biowaivers, and that's why it can avoid the time consuming, cost consuming clinical endpoint studies. Then it is critical for the complex generics like creams, gels, ointments and lotions. So this is the introductory part. The video might go lengthy, but I request you to stay with me till the end of this video, so that in a single video you will have introduction to the topical formulation bioequivalence. Now, why topical bioequivalence matters? So it ensures therapeutic equivalence of the generics with the reference products, because reference products are coming into the market after complete clinical studies, and generic formulations are not as studied as per the reference products. So how to ensure the therapeutic equivalence? So it is by showing the bioequivalence. The bioequivalence may be by the in vitro studies and maybe by in vitro plus in vivo, or maybe with the in vivo as per the case by case basis. So topical bioequivalence matters as it can avoid the costly clinical endpoint studies whenever possible. It supports the patient access to affordable medicines, and it can minimize the risk due to differences in the formulations and the skin permeation. So to show the therapeutic equivalence, the bioequivalence is necessary. Now coming to the regulatory pathways. So as per the market, as per the country specific requirements, the regulatory pathways may have some differences and may have some similarities. So consider two markets, regulated markets like USA and the Europe. So for USA it is US FDA, for Europe it is EMA. For US FDA, it emphasizes on the in vitro and in vivo performance based methods. And there are initiatives from the GADUFA to reduce the reliance on clinical endpoint trials, because clinical endpoint trials are time consuming and costly. As per EMA, guidances on locally acting products and topical BE, and other agencies are making the similar effort. Now coming to the experimental approaches, so you might have heard the terminologies like IVRT and IVPT. IVRT and IVPT are the in vitro measurements, methods or experimental approaches to demonstrate the similarity between reference and generic. IVRT, that is in vitro release testing, measures rate of drug release from dosage form, useful for detecting formulation differences. In vitro permeation testing, that is IVPT, it uses the human, animal or animal skin models and it studies the drug permeation. In IVRT, the drug release is tested, and in IVPT, the drug permeation is tested. Then coming to the in vivo methods, these are the dermal pharmacokinetics, microdialysis, microperfusion, and clinical endpoint studies. So dermal pharmacokinetics, these studies are called as tape stripping, quantifies drug levels in the stratum corneum. Microdialysis or microperfusion measures the drug concentration in the dermis or epidermis. So depending on the penetration, these studies are performed. Then clinical endpoint studies, these are less performed, these are highly time consuming, cost consuming, and these may be variable. Now coming to the challenges in the topical B. So as the formulation is topical, there are different variations for the bioequivalence. First is the skin is a heterogeneous and variable barrier. Different subjects skin will differ, and that may be a challenge. Formulation complexity is there. Depending on to the formulations, some are emulsions, suspensions, semi-solids. In some formulation, the drug is dissolved form. In some of the formulation, it is in the dispersed form. In some of the formulations, you may get the some amount of the API dispersed and some amount dissolved. Then lack of universally accepted surrogate endpoints, need for the standardization of the methods and harmonization across the regulators. It is very important and it is a challenging aspect. Evolving the scientific tools, advanced analytical techniques like confocal Raman spectroscopy, imaging, mass spectroscopy, model based approaches are there, which are evolving day by day. PBPK modeling is there. PBPK means physiologically based pharmacokinetic modeling. It can be used for the skin absorption studies and integration of the QBD principles for the generic topical design. Regulatory considerations, as I have mentioned, sometimes the in vitro testing can suffice the requirement, sometimes both are required, sometimes Q1, Q2, Q3 plus in vitro testing is required, sometimes all are required, sometimes clinical endpoint study is must. So depending on to the formulation, depending on to the indication, depending on to the formulation, the regulatory considerations can be different. So acceptance of the IVRT, IVPT plus dermal PK as alternative to the clinical trials. Importance of Q1, Q2, qualitative, quantitative sameness in the formulation. Q3 sameness, that is microstructure equivalence, are critical for the topical formulations or semi-solid formulations. And agencies are moving towards weight of evidence approaches. Weight of evidence approaches, that means how much proof is there for meeting the therapeutic equivalence. Now coming to the conclusion of this video, we need to understand about the requirements from regulatory point of view, from quality point of view, from formulation point of view. Then in the topical formulations, scale up, site transfers can add some more difficulties. Demonstrating topical bioequivalence is complex, but it is evolving day by day. Combination of in vitro, in vivo and modeling approaches can provide you the robust evidence of meeting the therapeutic equivalence. Regulatory flexibility is also increasing to support the generic innovation so that the public can get the quality medicines in a cost effective way. So considering the topical formulations, not only the in vitro studies, but also the modeling, but also the mechanistic understanding about the products and using of the PBPK principles can be useful. So if you are working onto the topical formulations, always remember and understand the requirement of demonstration of bioequivalence, because these are the semi-solid formulations and the release of the API from these formulations is dependent onto the product, depending onto the excipients, and these are not the solution formulations because these are semi-solid, these are applied onto the skin. There are various variations considering the composition, considering the Q1, Q2, Q3 sameness, release and permeation. So always have deep understanding about the product, about the requirements. Thank you for watching the video and let's meet soon with another informative video.